MS2-Affinity Purification coupled with RNA sequencing, or MAPS, has been developed to identify the whole targetome of any RNA of interest in bacteria. This technology makes it possible to identify any kind of sRNA partners independently of the mechanism of regulation. The characterization of sRNA-dependent regulatory networks will help understand better virulence factors production, biofilm formation, and survival of the bacteria within the host cells, and ultimately fight against staphylococcal infections. This protocol can be applied to any pathogenic or non-pathogenic gram-positive bacteria. Demonstrating the procedure will be Noémie Mercier, a PhD student from Dr. Pascale Romby's laboratory. To begin, grow one colony of strains carrying either PCN51P3MS2 sRNA or PCN51P3MS2 plasmids in 3 milliliters of BHI medium, supplemented with 10 micrograms per microliter erythromycin in duplicates. Dilute each overnight culture in 50 milliliters of fresh BHI medium supplemented with erythromycin to reach an OD600 of 0.05. Grow the cultures at 37 degrees Celsius with shaking at 180 RPM for 6 hours. Transfer each culture into a 50 milliliter centrifuge tube and centrifuge the tubes at 2,900 times G for 15 minutes at 4 degrees Celsius.